So I'm aiming today to present to you a project on the identification of proteomic clusters in the CSF of sporadic ALS patients. And in fact, this project is part of a bigger one that all started in the brain and got recently published in Nature Communication. So I already presented this study last year. Briefly, we collected 100 brains from ALS and control patients and also from a mice model of the four main proteins involved in ALS. And from these human and mice, we obtained different omic analyses ranging from genetic, RNA expression and proteins expression, aiming to identify and validate few targets linked to ALS disease. But one other key element of this publication was that when focusing on RNA expression of only ALS patients, we were able to identify clusters. And what was different between the cluster, here you can see we identify with hierarchical clustering at least 1, 2, 3, 4 different ALS type. And what you can see is that we obtain different expressions, so blue is low expression and red high expression of genes associated with immune response, but also extracellular matrix, and also different range of synaptic genes that were either highly expressed or lowly highly expressed. So it became clear from us that we can find molecular cluster of ALS, they can be defined, and also logically, if different pathway alterations are observed in these patients, this might result in different response to medication, right? But then, since it's not possible to take the brain from patient alive, we then went for analysis in the CSF, where we collected from around 100 patients, extracted protein and analyzed them with mass spectrometry. The first step of this study was in the context of biomarker validation to identify proteins and pathways that would be specific to the disease in general, but also that would be sex-specific. So what happened in males with ALS, what happened in females, is it the same or not? In the second step, we aimed to determine if there were also clusters that could be defined from CSF sample, and if yes, to characterize them with proteins and pathways. So when we do omics analysis, proteomics, this is kind of the result you have. You have like a heat map showing the different abundance of the proteins. Here we organize all the ALS patients on the right side and the control on the left side. At first see, just looking at the 600 protein, you cannot see obvious changes between ALS and control. Okay, then we also collected a lot of demographic and clinical information for mainly ALS, but also some for control. And I think one striking thing is that just looking at this information, we were not able to see a cluster of clinical demographic that would correlate with a specific molecular pattern. So patients with the same clinical information would rather not have the same molecular pattern. So here also a same treatment can result to different response in the same clinical pattern. So now let's focus by comparing ALS and control. Also on this PCA, you can see that at first see again, we are not able to clearly separate ALS and control. However, by doing differential expression analysis, you can see that we observed 47 proteins that were more abundant in ALS versus control, and 55 that were less abundant. What was interesting is to see that the chitinase family here was indeed higher expressed in the ALS patient compared to the control, which is proof of concept of our analysis here. Looking at the pathway that were mainly altered in ALS versus control, we identified change in immune response and also body fluid and coagulation, mainly by downregulation of fibrinogen, coagulation proteins, but a lot of immunoglobulins and apoproteins, for example. In contrast, we find increased activity of neuronal function pathways and also metabolic and catabolic process, with example of the prion-like protein, oligonucleotide-milling glycoprotein, or aldolase or fucosinase proteins. Now, when we focus at the sex-specific alteration, we stratified these results by male and female. And when you can see that analyzing male only, we were able to identify, for example, the prion protein again, aldolase, chitinase, complement factor, same as in the global cohort. We were not able to identify any signal in the female. However, if you look at the chitinase here, so you can see like from the global cohort in red, male only, which is both significant. You can see that in female it's still highly increased, but it's just not, it doesn't come out, so it's just not significant. So I would say that we have kind of similar signal in male and female, but it's just like decrease in the female. In a second step, we aim to identify if we could determine cluster from the CSF, and therefore we trained 4 different algorithms, testing if we can have cluster between 2 and 10, which one would be the best. And then we perform different analyses to determine the stability and the robustness of the clustering algorithm. To make a long story short, we were able to determine that the best analysis would be like using the K-mean algorithm and doing 2 or 3 clusters with our cohorts. On the right side here, you can see the results of the clustering for 2 cluster and 3 cluster. And what is interesting, so here you can see the number of patients when splitting into cluster or number of patients splitting in 3 cluster. And what is interesting is when you split from 2 to 3 cluster, we can see that some of the patients kind of remain stable here, that we call the alpha group. On the opposite, we find other patients that stay stable in the beta group, and we have some patients that are intermediate and then found in between and will be called the theta group. But to understand what's the alpha patient or beta patient, let's focus a bit more about the K-mean 2. So that's the result of the 2 cluster analysis. Here you can see the partition of patients either in the alpha group or the beta group, so this time they are well differentiated. And we perform here a differential expression analysis comparing alpha versus beta and found a bunch of proteins that were differentiating these 2 groups. You can find a summary of this analysis on this heat map, with each column here being a patient and each line being a protein that would be specific to alpha group or beta group. So what is an alpha and a beta patient? Here in the alpha group, you can find mainly activation of immune response pathways, and for example, also body fluid and coagulation, with high expression of fibrinogen, coagulation protein, with immunoglobulin, and for example, the aptoglobulin-related protein that I show here as an example that is highly increased in alpha compared to beta significantly. In contrast, a beta patient would be more characterized by high expression of neuronal function and cell junction and adhesion, with increased expression of the, for example, the cadherin, collagen protein, but also like receptor associated with synaptic and axonal elongation, such as the PTPR and type N receptors. Yeah. So in conclusion, with this analysis, we were able to define 2 clear clusters. One ALS cluster that is linked to immune response and coagulation, and the other that is more associated with high expression of neuronal function and cell junction and adhesion proteins. Now focusing on the 3 cluster analysis, you can see here when we split patients in 3 clusters, how they are separated. We again did differential expression analysis, but here we had to be careful that we had to compare each group to the 2 other, and we only selected protein if they were significantly different from the 2 other groups. So this is the summarized here on the right side, where you again see the patient as a column and the protein that is specific of the group as a row line. So in the alpha group, we were able to identify 86 proteins that were specific to this group. And if you can see, we found again like complement proteins, fibrinogen, and we found again this HRP protein that is highly expressed in alpha, but not in the 2 other groups. So it's mainly again a group that is associated with immune response and coagulation, same as the 2 cluster analysis. The beta group here, we found a protein associated with collagen, for example. We have again this PTPRN protein that is highly expressed in beta, but not in the 2 other group. So we mainly found that this group was again associated with neural function and cell adhesion. And finally, this intermediate group here had some expression of proteins, so 8 were specific to this group, with some of them that are chemokine, so associated with immune response. We found some cadherin, and we found also LINX1 that is binding to the nicotinic acetylcholine receptor, for example. So kind of a cluster that is in between and have activation of both pathways. So whatever, if we do a 2 cluster or 3 cluster analysis, what looks clear to this cohort at least is that we have a clear immune response and coagulation cluster and a neuronal function and cell junction adhesion cluster, so 2 different patterns of ALS patients. Finally, we try to correlate our groups, molecular subgroups, with the clinical information that I showed you at the beginning. And what we were able to observe is that the alpha group, the one that is linked to immune response and coagulation protein increase, had a significant decrease of age at onset for the disease compared to the beta group. So they developed disease earlier. This we were able to observe for the 2-cluster analysis, but as you can see here also for the 3-cluster analysis. Alpha had younger age at onset compared to beta, but also theta. So in conclusion, the immune response and coagulation cluster could represent a younger age at onset ALS population. So as conclusion, I would say that this study identified CSF profile of ALS patients. So ALS patients show sex-specific differences with decreased signal in female compared to male. We also observed molecular heterogeneity with kind of 2 or 3 main clusters in the CSF of ALS. So one cluster could be called immune response and coagulation. Another cluster was identified with increased neuronal function and cell junction adhesion. And we also observed an intermediate cluster, which is not clear if it's just 2 clusters or really a third one for now. Also, the immune response cluster show younger age at onset compared to the other clusters. And what is highly interesting in this study is that, I guess, it showed that CSF-based subgroup could contribute to patient stratification and personalized clinical trial. Of course, validations are ongoing on a new cohort of 100 patients, and we hope to validate this data. So this is just my group from Munich and all the collaborators on the project. Thank you.
Thank you, Laura. We'll take a very quick question. Yes. Luc.
Very nice talk. Did you try to explore your data set regarding TDP43 cryptic peptides, for instance, and also whether your proteins of interest in the beta or alpha clusters might be regulated by cryptic splicing at the gene level, I mean by TDP43?
So no, because here in the CSF we don't have transcriptomic data. So
you could explore. We have maps of cryptic exons, so maybe you can just try to benchmark whether your regulated peptides are actually, for instance, the downregulated ones could be targets of TDP43.
Oh yeah, this is definitely something we could check, and maybe I could discuss that with you later too. Yeah, we'll discuss. Yeah, thanks.
I have 2 short comments about the CSF proteomic. About the ALS patients, it is sporadic ALS or with the base genetic known or unknown genetic cause?
So it's mainly sporadic patients. So they come to the clinic and are diagnosed having ALS, but they also do genetic testing, and we found 3 of them that have C9 mutation. But these patients do not cluster together. They don't look to have a specific protein patterns. So they are kind of spread in the heat map.
Okay, so it's a mix of, sporadic.
It was sporadic, so they don't have history of familial ALS.
Oui.
But some of them we found. So they had
some of them. Okay.
3 of them have a mutation,
yes,
but not.
My second question is about when you separate the male and female in the CSF analysis. Did you perform the comparative analysis with control, female and male control?
Exactly.
Because you showed only control and male and female ALS, but not male and female control.
So I each time compared the female ALS versus the female control and the male ALS versus the male control.